Welcome back. In the following recordings, we will cover trematodes. As a reminder, we are still in the big category of helminths, which are the worms. And so far, we've talked about the nematodes, which are the roundworms. Now we're covering the trematodes, which are the flukes. And the final chapter for the helminths will be the cestodes, which are the tapeworms. So again, we are in the phylum of platyhelminths, the flatworms. And there's two classes here, the trematodes and the cestodes. The following bullets on this slide and the next slide are general characteristics of the flukes. And I will point out the unique ones which usually belong to the blood flukes, the schistosomes, as they come up. These recordings are set up a little bit different where I'm covering general characteristics, pretty much the life cycle of all the flukes at once, and then we'll talk about the individual organisms categorized by where they live in the human body. So, to get started, all of your flukes are flat, leaf-like organisms. So if you remember, the roundworms had round shape for their cross-section. These adults are flat, leaf-like organisms. Also, all the adults have two cup-shaped muscular suckers. So that's how they attach to our intestines or our blood vessels or wherever the adults live. For the most part, most of your flukes are hermaphroditic. That's what goes in this blank, meaning the same organism contains male and female reproductive structures. Your exception is the blood flukes. So the three species we'll talk about for the schistosomes, those are unique because they have separate male and female organisms. They are non-hermaphroditic. And for all the flukes, the common names will indicate where the adults live in humans. Now, compared to the nematodes, the life cycle has become increasingly more complex for these guys, and I'll show you in a second. But all the eggs must reach water to mature, so that's unique for all of the flukes. And all of the flukes have a common first intermediate host, which is the snail. And if you remember from your definitions, the intermediate host is where the larval stage or the asexual reproductive stage occurs. So for all of them, it's a snail. There has to be a snail involved for all these organisms to continue their life cycle. And then man is the definitive host, meaning where the adult and or sexual reproductive stage occurs. Now I will never ask you about geographical distribution, but I think it's interesting to note where these guys live. So for the most part, we don't see them that much here. They're more far east, like Asia for the Orient, and then Africa, South America, and the Caribbean. Okay, the infective stage for most of our flukes is called the metacercaria. And when the metacercaria is the infective stage, we ingest that in raw or undercooked water animals or vegetation. For the blood flukes, the infective stage is the cercaria. And we have to get infected with that by actual cercarial skin penetration, meaning we do not ingest the cercaria. It gets punctured through our skin, much like direct penetration with the hookworms or whatever when we talked about the nematodes. So lots of new terms here. We've got a metacercaria stage and a cercaria stage. Now the diagnostic stage for all your flukes is eggs, but depending on which organism we're talking about, it could be eggs in feces or eggs in sputum or eggs in urine, but it's always the egg stage for your flukes. Okay, so now I'm going to categorize the organ-dwelling flukes, meaning not the blood flukes, um, by where they live in humans. So you have 3 intestinal flukes, meaning the adults are going to inhabit the intestines for these 3. So there's Fasciolopsis buski, also known as the giant or large intestinal fluke, Metagonomus yokokawai, and Heterophyes heterophyes. Metagonomus yokokawai and Heterophyes heterophyes do not have common names, but you do need to know the common name for Fasciolopsis buski, which is the giant or large intestinal fluke. Then we have 2 liver flukes, meaning the adults of these 2 guys are going to inhabit the liver or bile duct area. So there's Fasciola hepatica, also known as the sheep liver fluke, and Opisthorchis sinensis, also known as the Chinese liver fluke. Now, Opisthorchis sinensis is also known as Clonorchis sinensis, and that's Clonorchis. And I mention that because if you're reading a different textbook or if you're looking on the CDC website or whatever, another place, they might mention Clonorchis sinensis or Opisthorchis sinensis. These 2 genuses are used interchangeably, so it doesn't matter which one you want to use. Some people might have already taken a parasitology course and you learned Clonorchis sinensis, so that's fine. For me, you can use either term. I am going to use Opisthorchis sinensis. And then we have 1 lung fluke, and that's Paragonomus westermanni, and its common name is the oriental lung fluke. Now, I've mentioned these schistosomes several times. These are your blood flukes, meaning the adults are going to inhabit blood vessels. They're not inhabiting tissues of organs. They actually live in the blood vessels. So the genus of all of these is Schistosoma, and we have 3 different species. Schistosoma mansoni, also known as Manson's blood fluke, Schistosoma hematobium, also known as the bladder fluke, and Schistosoma japonicum, also known as the oriental blood fluke. And the schistosomes are going to be unique for several reasons. They're sort of the black sheep of the fluke category. So they have lots of differences as compared to the other flukes. So we'll talk about those after we talk about all the other ones. Okay, I know this looks like a busy picture, but it's actually very simplified, and it's important because it's putting all the flukes together in one picture for you, except the blood flukes. But I'll mention that when it comes up. Okay, so this is your life cycle for the liver, lung, and intestinal flukes. So let's talk through it. So at the top, eggs are getting shed from humans. So it could be eggs in feces or eggs in sputum. Which one already do you think we're going to get eggs in sputum from? It was our 1 lung fluke, which was Paragonomus westermanni. So remember, all the eggs have to reach water to mature. Out of the egg comes a life cycle stage called the miracidia, or miracidium would be singular. And the way this gets out is by the operculum. So I've mentioned this before in a previous lecture when we talked about concentration techniques. So an operculum is a structure on some of your eggs that's like a little door or hatch or a little lid that can come open when it gets into the water, and this is how the next stage is released from the egg. So the miracidia get released when the operculum opens up. Now, to refresh your memory, when an egg has an operculum, what concentration technique is better to recover those eggs? So we had flotation and sedimentation. Remember, sedimentation is better to recover operculated eggs because if you use flotation, the operculum could open up and it could sink. It could cause the egg to sink to the bottom, and then you wouldn't be able to recover the egg at the top of the test tube when using the flotation technique. Okay, back to the picture. So the miracidia is actually the infective stage for the snail. So remember, the snail is your first intermediate host for all the flukes. There has to be a snail involved. So the snail gets penetrated by the miracidia or actually ingests it. So within the snail, there's actually several more life cycle stages that occur, and I won't make you learn all of those. But what happens is that a stage called the cercaria or cercariae emerges from the snail. And just one miracidia can result in hundreds of cercaria being released. So then we have this life cycle stage called the cercaria. Now, the cercaria could actually go and live in fish. It could get eaten by fish. It could get eaten by crabs or crawfish, or it could attach itself to water vegetation. Or for the blood flukes, which isn't being pictured here, this is actually the infective stage for the blood flukes, is that humans get skin penetration by the cercaria. So that could be another little arrow going down. But in any case, when the cercaria either attach to water vegetation or get eaten by fish or get eaten by crabs or crawfish, the next stage that comes from the cercaria is called the metacercaria. So these are like second intermediate hosts for the flukes, where it's got to attach or get eaten by something else, and the metacercaria develop next. So then we get infected by ingesting the metacercaria, either if we eat undercooked water vegetation, or we eat raw or undercooked fish, or we eat raw or undercooked crabs or crawfish. And that's how we get the flukes, is by ingesting the metacercaria stage. So we can get infected with Fasciolipsis buski or Fasciola hepatica by ingesting metacercaria on undercooked water vegetation. The way we get Opisthochus sinensis and Heterophyes heterophyes and Metagodamus yokogawai is by ingesting metacercaria inside undercooked or raw fish. And the way we get Paragodamus westermanni is by ingesting metacercaria inside raw or undercooked crabs or crawfish. And then remember, what's not being pictured is the way we get the blood flukes is by skin penetration of the cercaria. So, I know that seems like a lot, but I will keep saying it over and over again, and hopefully it'll start to sink in and make sense.